Hello everybody, how are you all? I think so you all are well and now becoming more serious towards your examination and the final revision courses. Okay, so myself Sunil Pawar from New Catholic Mission High Secondary School once again welcome you back for the new session of 12th class biology. Today we are going to start our new chapter, that is the 11th chapter, biotechnology, principles and processes. Clear? Now in this one basically this biotechnology chapter is divided into two parts. One, that is the principles, and second is the application of the biotechnology. Right? Now when we talk about the biotechnology then see that this word is made of two words. One, that is the bio, and second is the technology. Okay, you all are well aware about the technology. Okay, as you know that simply if you want to say that, then what is the technology? It is nothing special. It is just the what do you say, the use of scientific knowledge for the welfare of the human being. Okay, or if you want to say that, then we can use one more thing, that is the conversion of non-resource into a resource, that is called as technology. Clear? Simply the application of the scientific knowledge is simply called as technology. And when we talk about in the biological references, then we can say that this scientific knowledge which is related with the biology is used for the welfare of the mankind or the human beings. Clear? By using this different organism as a source is called as biotechnology. See that this term biotechnology was coined by Carl Erke in 1917, and this biotechnology is the techniques of using live organisms or enzymes from organism to produce products and processes useful to humans. That means nothing is special. Already these organisms are producing this one thing, but we are using this organism on a large scale to obtain their products. Okay, and these products are quite useful for the human beings. Okay, so you can see that this is just like that, the use of the antibiotics, okay, the vaccines, these all are very common facts. Now these days we are also using this biotechnology in the improvement of the qualities of the insertion of the new characters or the introduction of the new features in the living beings. Okay, so with the help of this one thing, whatever the products which already produces by this organism, okay, so by knowing them we are producing them on the large scale in a given medium of this one thing. Clear? Biotechnology is the integrated branch of biology which deals with the techniques of using live organisms or enzymes from organism in order to produce the different products and processes useful of human beings. Clear? Now these human beings have been using biotechnology for thousands of years. For example, the production of curd, cheese, bread, vinegar, wine, these all are done by the process fermentation. Is one of the oldest biotechnological process. And the many processes like in vitro fertilization, okay, leading to the test tube baby, synthesizing gene and using it, developing a DNA vaccine for correcting a defective gene, these are also the part of the biotechnology. Okay, just like that this fermentation, okay, now it is quite used on the industrial scale. Okay, and it gives such a great contribution in the industries and for the economical purposes too. Okay, now this in vitro fertilization, that means the fusion of the sperm and ovum outside the body. Okay, you know that this time because of this new lifestyle and the some physical problem, there are so many technical problems is observed in conceiving the pregnancy. Okay, so now with the help of this technological use or the application of this biological knowledge is quite useful for producing these babies, okay, by this kind of which one thing, the couples or the families, okay, and it just considered as a boon for the this kind of infertile or the less fertile couples, okay. Now they are also blessed by this baby with the help of this in vitro fertilization. In vitro means what? When the fertilization takes place where? Outside the body in a favorable condition in the laboratory, okay. While in vivo means inside or in the normal or the basic processes. Clear? Now, so here the European Federation of Biotechnology has given a definition of biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for the products and services. Clear? That means simply the use of the biological knowledge, the organism, and some tools like the enzymes and the genetic material for obtaining the different products in service of the human being. That is simply called as biotechnology. Okay. Now here we are going to discuss first, that is the principles of the biotechnology. When we talk about the principles of the biotechnology, then there are basically two facts. One, that is the genetic engineering, and second is the sterilization methods. Okay. So you know that this genetic engineering is especially the process. Engineering means what? Any kind of manipulation in the pre-existing content to make it more useful and modified in the nature. Okay, that is called as engineering. And when we talk about the genetic engineering, then any kind of manipulation which perform with the genetic material, either by insertion, either by deleting or introducing the new character, or by correcting the pre-existing defective genetic material. This all headed under which heading? That is the genetic engineering. Okay, so this genetic engineering is also a good tool for obtaining the desired character. Okay, it is also very helpful for correcting the pre-existing varieties. Okay, it is also helpful for improvement of the pre-existing varieties. It is important for obtaining the desired character in the pre-existing species. Okay, so this genetic engineering is actually the direct manipulation of the genome. You know that whether it is DNA or RNA of an organism, it involves the transfer of new genes to improve the function or trait into the host organisms and the changes the phenotype of the host organism. Okay, not only the phenotype, but also it is responsible to change the anatomical and physiological activities too. Okay, now the maintenance of the sterile conditions in chemical engineering process of enables, okay, growth of only desired microbes for manufacture of bacteriological products like antibiotics, vaccine, enzymes, etc. This under the sterilization method. Clear? This sterilization method is nothing special. It actually provide a quite hygienic conditions. Okay, a quite a beneficial condition for the accurate copying of the manipulated genetic material without interference of the surrounding. Clear? On the large scale. Clear? So this traditional hybridization used in the plants and animals breeding leads to the inclusion and the multiplication of the undesirable genes along with the desired traits. Okay, so the techniques of genetic engineering, which include the creation of the recombinant DNA and the use of gene cloning and the gene transfer, allow us to isolate and introduce only one or a set of desirable genes without introducing the undesirable genes into the target organism. Okay, you know that naturally whatever the genetic material is present, they used to copy themselves during their multiplication. Okay, but if there is any defective gene or any defective genome or any defective material, okay, then as a normal procedure it will copy it along with this one. Okay, so by improving this technique or by the execution of this technique, okay, we can remove this defective part and only be allowed to multiply the useful genes. Okay, only the such genetic material is used to multiply which is desired in nature. So this is one of the basic facts. In the chromosome there is a specific DNA sequence called the origin of replication, which is responsible for the initiation replication. Okay, so therefore the multiplication of any alien piece of DNA, what is the meaning of alien piece? Which is not the part of that genome or that genetic material in an organism. It needs to be a part of a chromosome which has a specific sequence known as the origin of replication, and thus an alien DNA is linked with the origin of replication, so that this alien piece of DNA can replicate and multiply itself in the host organism, and this is known as the cloning or the making multiple identical copies of any template DNA. Okay, see that. Two basic words which we want to explain here. One, there is the origin of replication, and second is the alien DNA, and finally this term which you are quite familiar, that is the cloning. Okay, so first of all, what is the alien DNA? The alien DNA is such desired DNA which we want to introduce in the pre-existing genetic material. Okay, and for this procedure we need this origin of replication. Actually, what is this origin of replication? It is a specific sequence of the nucleotides, okay, or this which arrange on this DNA sequence, in the DNA sequence. Clear? It is quite similar in this host organism's genome as well as that alien genome. Clear? So from it is compulsory to have this origin of replication. So from this one thing they used to cut and fix with each other. So this way a hybrid DNA will develop, and this hybrid DNA will further separated and only manipulate and motivated to clone itself. Okay, so this way a large number of copies is actually made there. Okay, so the construction of the first recombinant DNA emerged from the possibilities of the linking a gene encoding antibiotic resistance with the native plasmid of Salmonella typhimurium. Okay, so this time first time become possible with the help of this one thing. Okay, so this scientist, that is Stanley Cohn and the Herbert Boyer, in 1970 isolated the antibiotic resistant gene by cutting out a piece of DNA from a plasmid. You know that. What is the plasmid? It is the extra chromosomal, the circular small segment of DNA which is present in the bacterial cell, which is self-replicating, self-multiplying and self-inheriting in nature, okay, of the Salmonella typhimurium. Okay, the cutting of this DNA at a specific location become possible with the discovery of the so-called molecular scissors, that is the restriction enzymes. Okay, now their separation, then the addition. So first we need to cut them from that entire genetic material. Okay, or if it is plasmid, then we need to cut it because it is a complete ring shaped structure. Okay, so it become only possible when we discover this molecular scissor. What is this molecular scissor? It is not a mechanical tool, it is a biological tool which is called as a restriction enzymes. You know very well that there are two type of restriction enzymes are there. One is exonucleases and second is the endonucleases. Okay, exonucleases is responsible to cut the DNA from its outermost part, okay, or the ending parts, while this endonucleases is basically used to cut the desired point of the DNA which is present in between of these two external points. Clear? Now, so the cut piece of DNA was then linked with the plasmid DNA, and this plasmid DNA acts as the vector to transfer the piece of DNA attached to it. Okay, and a plasmid can be used as vector to deliver an alien piece of DNA into the host organism. Clear? So now this desired DNA is now attached with the plasmid, and this way we form a recombinant DNA, and because this is present in this bacteria, it is responsible to carry it to the host organism. That's why it is called as a vector DNA. Clear? And now this vector DNA is carried to the host organism where the copies of this will form. Clear? Now, the linking of the antibiotic resistant gene with the plasmid vector become possible with the enzyme ligase. It is also called as the adhesive, okay, which is responsible to join the two segments of DNA. Okay, it is also considered DNA ligase, and which acts on the cut DNA molecule and join their ends, and this makes a new recombination of autonomously replicating DNA created in vitro and known as the recombinant DNA. Okay, already we discussed that what is the recombinant DNA, but simply if we conclude this word from the this meaning from this word, then recombinant DNA, that means such DNA which obtained by the combination of the two different DNA is called as recombinant DNA. Now, when this DNA is transferred into E. coli, it could replicate using the new host DNA polymerase enzyme and make multiple copies. This is called as the cloning. Okay, and the ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. So whatever the recombinant DNA which we made is now transferred in the bacterial cell, and basically this E. coli is the most convenient source or as a host we are using in this genetic engineering. Clear? And when it added in the bacterial cell, then by using this DNA polymerase enzyme of the this bacterial cell, it used to multiply itself, and this way we can obtain the desired copies of the DNA in the required numbers. Okay, so this recombinant DNA technology is also called as the genetic engineering. Simply we can also call this genetic engineering as the recombinant DNA technology. Such technology which used to combine the two different DNA segment to obtain a hybrid DNA is called as recombinant DNA technology, and that hybrid DNA is called as recombinant DNA. Okay, simply it is the manipulation of the genetic material, so it is also known as the genetic engineering. Clear? Now, it deals about the production of the new combination of the genetic material in the laboratory, and this recombinant DNA molecules are then introduced into the host cells where they can be propagated and multiplied, as we learned in the previous point that how we can make the copies of this newly produced hybrid DNA. Now we are going to discuss the steps of the recombinant DNA technology. So first of all, identification of the DNA with the desirable gene. Okay, that means this is the desired gene which we needed. Now these days actually this desired gene can be obtained in three forms. Okay, one, that is the cDNA, that is the complementary DNA which we can produce as the complementary threads of the DNA. Clear? Second is the asDNA, which we call that the synthetic DNA. Okay, what is synthetic DNA? Which we produce artificially where in the laboratory, that is called the synthetic DNA, right? And third one, that is the rDNA, that is the randomly produced DNA molecule. That means in this one we are not synthesizing, we are not copying, but we are cutting the different segments of the DNA from the different DNA molecules, and by using this different enzymes, that is the ligase enzyme, we used to make it single thread, and by using this complementary or the semi-conservative method, we can develop it the other strand too. Clear? Now, so this desired DNA can be obtained by this three method. Then the introduction of the identified DNA into a suitable host to form the recombinant DNA. So this desired DNA is need to attach with a suitable host to form this recombinant DNA. And then the maintenance of the introduced DNA in the host and transfer of the DNA to its progeny. So this is the production and transfer to the next generation. Clear? Now then, what are the tools is actually needed for the recombinant DNA technology? Clear? In this one, the first thing, that is the most important tool, that is the restriction enzyme. As we discussed earlier, what is the restriction enzyme? Then the polymerase enzyme, which is responsible for the polymerization of the nucleotide for the formation of a long segment of DNA. Then this ligase, it is considered the adhesive. Okay, then the vectors means what? One who used to carry them from one generation to next generation, or from one organism to the host organism. Clear? And finally the host organism, the suitable microbe which we need to multiply the copies of this hybrid DNA. Okay, so one by one we are taking this one thing. The restriction enzymes, it is the molecular scissors. Okay, as I told you, these are the two, one that is the exonucleases and second is the endonucleases, which basically used to cut the DNA of the desired DNA segment or the that plasmid. Okay, that is called as the exonuclease and endonuclease from the inner point or the external surface. Is clear? Just like that, the first restriction endonucleases, that the in second, always cut DNA molecules at a particular point by recognizing the specific sequence of the 6 base pairs, and the specific base pair sequence is known as the recognition sequences for in second. Clear? And each restriction endonuclease restriction specific palindromic nucleotide sequence in the DNA. Clear? Remember, this endonuclease enzyme actually responsible to search a specific series of 6 nitrogenous base pair, or what you call the nucleotide, on the DNA segments. Okay, and this segment is called as the palindromic nucleotide sequence. Clear? And when by identifying it, it used to cut it. And these palindromes are group of letters that form the same word that read both the forward and backward. For example, Malayalam, M-A-L-Y-A-L-A-M. Similarly, M-A-L-A-Y-L-A-M, Malayalam. Clear? So similarly, here if you observe, then one of this palindromic sequence is observed. See that this is the 5 to third end and this is the third to 5 end. Clear? So if you move from this 5 to third end, then you will get GAATTC. This is the arrangement of 6 nucleotide. And when you come in the complementary or what is the second, it's complementary the strain from 5 to third side, then again you will get the reverse arrangement, GAATTC. Here is also GAATTC, here is also GAATTC. So this enzyme, that is the restriction endonuclease enzyme, is responsible to identify this palindromic sequences of the nucleotides in the DNA and used to cut it. Clear? So the palindrome in the DNA is a sequence of base pairs that reads same on the two strands when orientation of the reading is kept the same from both sides, from 5 to third end and 5 to third end, then it will is in the same one. Clear? Then this restriction enzymes cut the strands of DNA a little away from the center of the palindromic side between the same two bases on the opposite strands having the sticky strands. Okay, and this thickness of the strands facilitates the action of the enzyme, that is the DNA ligase. It is helpful for cutting, and then later on it also assists the addition of these two one. Clear? The restriction enzyme nucleases, endonucleases are used in genetic engineering to form the recombinant molecules of DNA, which are composed of DNA from different sources of genome. Okay, as I told you. And then when cut the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends and can be joined together using the DNA ligases. With the help of this diagram, you can see that this is the enzyme of this is the genetic material of the vector DNA, and this is the foreign DNA. Foreign DNA means what? The desired DNA which we require. Okay, so you can see that is the GAATTC, here is the GAATTC. Similarly, here is also GAATTC. So first of all, the breakdown of this hydrogen bonds is there and these two get separated. Okay, and then what happened? As I told you, not the complete one, but it cut in such a way where it has this free end. Similarly, AATT and here is AATT. So then this 2-1 is used to combine. Okay, this extended part of this palindromic site is used to combine with this one. Okay, and it is called as the addition site of the DNA segment, and it is basically need to combine and form this recombinant DNA, and this combination or this addition is performed with the help of the DNA ligases. Okay, now Moving forward, here it was actually technical, and here we can see that complete recombinant DNA technology, how it actually works. Clear? So see that here this is the foreign DNA, okay, which basically we obtain by the uses of the restriction endonuclease enzyme, and this way we obtain this specific desired part, or what you call that is a foreign DNA, or you can also call it the alien DNA. Clear? This alien DNA of, see that earlier this was the complete DNA part, okay, then by using this restriction endonuclease enzyme, we used to cut this specific DNA segment. At the same time we are using this plasmid, okay, and the use of this restriction endonuclease enzyme, which is responsible to read the same palindromic sequence in this desired DNA, and this one thing is used to cut this one, okay, and then now this desired DNA is introduced in this plasmid, and here we obtain a specific recombinant DNA molecule, which is a hybrid, which is a combination of this plasmid DNA and this alien DNA, okay, and then now it transferred to the host cell, that is here the bacterial cell E. coli, okay, and now here this E. coli when multiply itself, then but natural this recombinant DNA is also multiplied, okay, or if we need to make it more required copies, then only by using some stimulants we can motivate this specific DNA segment to multiply itself and form the copies of this one thing, cloning, okay. Now then here the separation and the isolation of the DNA fragments, whatever the DNA fragments which you obtain from this desired DNA molecule from the genome, so how it can be separated and how it can this fragment be obtained. Clear? So this fragment of DNA obtained by cutting DNA using the restriction enzyme is separated by the technology called the gel electrophoresis, okay. In 11th class we learned that what are the different tools which we use in the biology. So in this one thing, this gel electrophoresis is one of the most important tools, okay. So now this negatively charged DNA fragments can be separated by forcing them to move toward the anode under an electric field through the medium. So this DNA fragment separated according to their size through sieving effect provided by the agarose gel, okay, and the separated DNA fragment can be visualized after staining the DNA with that is the ethodium bromide, okay. What is this one thing? This is the which one thing? That is the radio label, okay, that means the radioactive material, okay, which followed by the exposure to UV light, okay. When this DNA segment is treated with this and ethodium bromide, and when we observe under the UV light, then it can be clearly seen and exposed, okay. So the separated bands of DNA are separated from the agarose gel and extracted from gel, and this is called as the elution, okay. This elution is nothing special, it is just the obtaining of the desired DNA segment from this agarose gel, okay, and the DNA fragment purified this way is used for the recombination. Clear? So earlier what is the use? You need to dissolve the outer cell wall or the cell membrane, then the nuclear membrane, then the that genome is used to cut or dissolve by this using the restriction enzyme. So you have so many DNA segments, okay. So here this electrophoresis plate is used where we made more than one well, okay. In this well, by using that micro pipette we added this fragmented DNA in this one thing, okay. Here cathode and here is the anode, okay, and when we force to move them from cathode to anode, then what do you observe? According to their gravity, according to their size and according to their charges, they will move, okay. So at the side of the anode you will obtain the more smaller part DNA segment, and this side you will obtain the heavier and the larger DNA segment. Clear? So these segments which we basically needed can be further labeled with the help of this ethodium bromide, and then under the UV light we can observe this one thing and can be separated from this one thing. So this is the separation and the isolation of the desired DNA. Okay, now then here we are going to move toward the cloning vector. Clear? This plasmid and bacteriophages, actually the plasmid, phasmids and cosmids, these are, so here we are marking them with the help of this, and this transformation is a procedure through which a piece of DNA is introduced in a host bacterium, and generally the genes encoding resistance to antibiotics such as ampicillin, chloramifenicol, tetracycline, or what you say, that is the kanamycin, these are considered useful selectable markers for E. coli, which actually restrict the multiplication of the other part of the DNA. Now next one, that is the cloning sites. So to link the foreign DNA, the vectors need to have single recognition site. For the commonly used restriction enzymes is the presence of more than one recognition sites within the vector will generate several fragments, and which will complicate the gene cloning. So the ligation of the foreign DNA is carried out at a restriction site presence in one of the two antibiotic resistant genes. Okay, so we only need to aid, and we only need to which one thing, that is the multiply that is specific segment. So by using this antibiotic resistance genes, we can only multiply and make the specific site of this gene. Clear? Now, so here with the help of this E. coli cloning vector, you can see that this one thing, E. coli RI, Clay1, Hin3, then H1, Cell1, okay, and here is this one. These all are actually which one thing, that is the restriction sites. Clear? While on the other hand, if you observe this one thing, origin, then the row, then amplification R, and this tat R, these all are actually the proteins involved in the replication of the plasmids. Clear? So remember, these are the antibiotic resistant site, and these are the which one thing, restriction endonuclease sites, which is basically used to identify and cut the segments. Okay, now then here is the insertional inactivation. What is this one? So the most efficient method of the screening for the presence of the recombinant plasmid is based on the principle that the clone DNA fragment disrupts the encoding sequence of a gene, and this is termed as the insertional inactivation. Okay, you know that basically the fragments which is responsible for disrupting the cloning, okay, which is responsible to multiply the sequence of the gene, is considered insertional inactivation. Just like that, an example which make you very clear that the powerful method of screening for the presence of the recombinant plasmid is referred to as the blue-white selection. Remember, this is the example which make you very clear that this method is based upon the insertional inactivation of the LEC-Z gene. Okay, you just recall the leucoperon. Okay, now LEC-Z gene present on the vector, then this LEC-Z gene encodes the enzyme beta-galactosidase, which can cleave a chromogenic substrate into a blue colored product. Okay, so if this LEC-Z gene is inactivated by insertion of a target DNA fragments into it, the development of the blue color will be prevented, and it gives white colored colonies. That means the production of this beta-galactosidase is restricted there. Okay, now so by this way we can differentiate a recombinant and the non-recombinant colonies. So which is a recombinant can be seen with the help of the product, and which is non-recombinant cannot be seen with the help of this absence of this product. Clear? Now then the vector for the cloning gene is plants and animal. There is Agrobacterium tumefactions. Okay, this is the pathogen of the agar plant, is able to deliver a piece of DNA known as tDNA, transform DNA, to transform normal plant cell into a tumor and direct this tumor cells to produce the chemicals required by the pathogen. Clear? These retroviruses in animals have the ability to transform normal cell into the cancerous cell. You know that this retrovirus is responsible to reverse transcription, and basically a non-malignant or the non-cancerous cell can be converted into the cancerous cell. Okay, so this tumor inducing a plasmid of Agrobacterium tumefaciens has been modified into the cloning vector, having no more pathogenic to plant, and similarly this retrovirus have been modified into the cloning vector for the animals. So these are the good option for this one thing. Then this competent host. What is this competent host? For the transformation of the recombinant DNA. Okay, so first of all, the simple chemical treatment with the divalent calcium ions increases the efficiency of the host cell to take up the DNA plasmid. Okay, so first of all you need to treat them with the divalent calcium ion. Then this recombinant DNA can also be transformed into host cell by incubating both on ice, followed by the placing them briefly at the 42 degrees Celsius. It is called the heat shock, from 0 to 48 suddenly, and then putting them back on ice. So this enables the bacteria to take up the recombinant DNA. Just to receiving the recombinant DNA, we need to give them this shock heat treatment. Clear? From 0 to how much temperature? 42 degrees Celsius, and again bring back to the 0 degrees Celsius. Clear? Then in micro injections method, in this one how to imply the recombinant DNA is directly injected into the nucleus of the cell by using a glass micropipette. Just by injecting inside the nuclear membrane, it can be applied there only. Then the biolistics of the gene gun method. Okay, gun to shoot it. Clear? So it has been developed by introduce recombinant DNA into the mainly plant cell by using a gene or the particle gun, and in this method the microscopic particles of the gold or the tungsten are coated with the DNA of interest and the bombarded onto the cell. So first of all, that desired DNA segment is need to coated on the two particles, either gold or the tungsten, and then it will be bombarded on that cell in which we need to transfer. Okay, and the last method is used, that is the disarmed pathogens, okay, vectors, that is the Agrobacterium tumefaciens. Okay, so which when allowed to infect the cell, then the transfer the recombinant DNA into the host is observed. You know that it is a pathogen. This agrobacterium is used to, first we need to introduce this recombinant DNA in the agrobacterium, and when this agrobacterium tumefaciens is infected the host cell or whatever that plant or animal cell, then surely it will be, this genetic material will introduce in this plant or animal cell where it need to be further in presence. Clear? So the process of recombinant DNA technology which we learned earlier, first of all we needed the isolation of the DNA, that means the obtaining of the desired DNA, then the fragmentation of the DNA by the restriction endonucleases enzyme, then the isolation of the desired DNA fragment, then the ligation of the DNA fragment into the vector, that making the hybrid DNA, then transforming the recombinant DNA into the host, that is by using this further procedure, either by shooting by the gun or by using that agrobacterium infection or whatever the procedure will suitable there only. Then culturing the host cell in a medium at the large scale, and then this extraction of the desired product. Okay, so once when you introduce into the host cell, then this host cell will further produces its copy in the desired number, and then this product will be separated or the extracted with the help of the particular method. Okay, so now here this isolation of the genetic material, first of all again simply we can revise it. So this genetic material is isolated from the other macromolecules by using the enzyme, that is the lysozymes, okay, cellulase, chitinase, okay, these are basically lysozyme for dissolving the cell, that the capsule and the membranous structure, cellulase for dissolving the plant cell wall of the cellulose, chitinase is responsible to dissolve and destroy the chitinous covering, and then DNA that separate out can be removed by spooling. Okay, and this spooling is what the extraction of this one thing what we needed, which is remain as the what do you say, the spare part. Okay, so this RNA can be removed by the treatment with ribonucleases, whereas the proteins can be removed by the treatment with proteases enzyme. Clear? Now this cutting of the DNA at a specific location. Now is, first of all, this DNA is removed from the nucleus, and now it need to cut. So it is performing by the restriction enzymes and the agarose gel electrophoresis to check the progression of a restriction enzyme which is responsible for the digestion, and after cutting the source of the DNA as well as the vector DNA with a specific restriction enzyme to the cut out the gene of interest. That means only that specific part of the DNA we need to cut, okay, which we need to produce the recombinant DNA from the source of DNA. Clear? Then the amplification of the gene of interest using that is the PCR. Simply we call it the polymerase chain reaction, PCR. Okay, to get the multiple copies of the DNA or the gene of interest in the vector by using the set of primers and enzymes, DNA polymerases. Clear? So what is this PCR techniques? We can learn with the help of this one thing. That is these three basic steps. Okay, what is the first one? That is denaturation. You know that the denaturing of anything is called as the denaturation. The double stranded DNA is denatured by using the high temperature of 95 degrees Celsius for 15 seconds. Okay, and now each separated single strand acts as a template for the DNA synthesis. As you know that this nucleic material is also protein. Okay, and when we heated them above this 80 degrees Celsius, then they will lose their bonding and get separated. So this is the denaturing. Clear? Then second is the annealing. The two sets of the oligonucleotide primers are annealed, okay, hybridized, that means to the separated single strands, and this step is carried out at the slightly lower temperature 40 to 60 degrees Celsius. Okay, by denaturing we just separated the two DNA segments. Then this annealing is responsible to which one thing, separated a single stranded along with this primers. Okay, then the third one is the extension. The thermostable enzyme, that is the take DNA polymerase, is used in this reaction which extend the primers by aiding that denatured NTPs. Okay, that is the deoxynucleotides. Okay, triphosphatase, the complementary to those of the template DNA. Okay, that is with the magnesium ions is required as a cofactor for the thermostable DNA polymerases to just keep them in the constant condition. Clear? And these steps are repeated many times in order to obtain the several copies of the desired DNA. And this application of the PCR in recombinant DNA technology, sequencing DNA fingerprinting, early diagnosis of the infections, diseases is now very popular in a diagnostic methodology. Clear? Now, then what happened? This polymerase chain reaction can be observed here. Okay, so what is the first one? Denaturing, second is the primer annealing, and third is the extension of the primers. Clear? So what do you observe that? Here you can see that this is the normal DNA segment where this part is the desired DNA which we need to amplify. Clear? So first we need to give it a heat temperature, that means the denaturing at near about 95 degrees Celsius. Clear? It is responsible to cut this one thing. You observe this two different DNA segment. Okay, this is the DNA strand, fifth and third, two and get separated, and here we need to apply or aid this primers enzymes. Clear? This primers is responsible to work with this primerase, and it is responsible to make the complementary strand here and this complementary strand here, and you may observe this amplified DNA. That means from this single DNA you can observe the many copies, and this is called the amplification. So at least 30 cycles is made, so you may observe more than 60 copies. So again and again by repeating these processes we can obtain the millions and billions of the copies of these substances. Clear? Now then here we are having the insertion of the recombinant DNA. So what DNA which we already obtained, we need to insert into the host cell. Clear? So include making the recipient cells competent to the receive. It take up DNA present in its surrounding and the recombinant DNA bearing genes for the resistance to an antibiotic is transferred into the E. coli cell, and the host cell become transformed into the ampicillin resistant cell. Okay, just like that here we obtain this resistant cell which we already obtain as a desired one, and we already make it copy. Now it entered into the host cell. Clear? Then obtaining the foreign gene product. Clear? So the foreign gene DNA multiplies in plant or animal cell to produce the desirable protein, and the expression of the foreign gene in host cell involves optimized condition to obtain, okay, then the recombinant protein, and the recombinant cell is multiplied in a continuous culture system in which the used medium is drained out from one side with fresh medium is added for the other to maintain the cells in their physiological active phase. That means the utilized and the already tired and worked out cells is need to remove, and the fresh cells need to inserted in this one thing. Clear? So a bioreactor provides the optimal conditions for achieving the desired products by providing optimum growth conditions. Okay, just like that, the temperature, pH value, substrate, salts, vitamins and oxygen, it all is basically maintained and provided with the help of a reactor which is called as the bioreactor. Okay, it is actually needed to maintain a specific condition there only. Clear? Now so see that these two bioreactors are shown here. One that is the simply stirred tank bioreactor, and second is the spurged stirred tank. Clear? In this simple stirred tank you can see that this bioreactor is actually quite large vessel which is having 100 to 1000 or 2000 liter capacity of the volume according to our requirement. Okay, here you can see that here in the middle there is a shaft. Okay, this shaft is having this flat blade impeller. When it moves, then it is responsible to rotate and shuffle, okay, whatever the material inside it. Clear? And in this one, from this side we can add this acid or base to maintain the pH. If medium is acidic, then you have to add this pH, and if medium is this base, and if medium is basic, then you need to add this acidic content. Okay, then here we have to insert this steam, okay, forcefully, just to keep it sterilizing in nature. That means not allowed to grow the other ones. Clear? And here this foam breaker is there because of its movement, that is the churning. Okay, a foam comes on the top, so this foam breaker is responsible to resend this material inside it. Clear? And here you can see that this flat bladed impeller is continuously moving with the help of this shaft, okay, which is attached with this motor, is responsible to develop a froth. Clear? And here this sterile air is continuously added in this one to make it full of oxygen in nature. Okay, so that's why no any anaerobic product is produces. Clear? So this simple stirred tank bioreactor, what you observe with the help of this procedure, this is nothing special. It is just a facilitated tank which used to maintain the pH value, aeration, okay, and the removal and the addition of the fresh cell for the multiplication purposes. Clear? So this stirred tank is there. In this one you can see that nothing special, but here this gas entertainment is there. That means only see that this is the propeller is there, which used to insert the air forcefully, okay, and this rotor is there, okay. This rotor is responsible to shuffle or churning this one thing, okay. So that's why continuously movement of this one is responsible to increase the surface area for the oxygen transfer, okay, and this increased surface area for the oxygen transfer is always keep the cell active to react and multiply there only, clear. And that's why these two reactors are there, remember this two one, okay. Now finally we are going to discuss the downstream processing. It is nothing special, it just involves the processes that makes the product obtained ready for marketing, okay, and this process includes the separation and purification called as downstream processing. That means what is downstream processing? Just whatever the product which we obtain, we need to purify it and we make it ready for the marketing purposes, clear. And the suitable the suitable preservatives are added to it and sent for the clinical trial in case of drugs before the releasing to market for the public use, okay. So whatever the product which you obtain by this genetical engineering, or the biotechnology, okay, first of all it need to tested, clear. When you obtain it in the purest form, now it is ready to apply, then we need to first make it trial, okay. So first we applied it on the trial purposes, trial basis, and if we got the required success in their result, then only we make it common for the use for the public, okay. So first we need to apply it on the different trial level, clear. So this is all about today's discussion, okay. What we learn about the biotechnology, its principles, okay. So it is basically based on two facts, one that is the genetic engineering, and secondly its amplification by obtaining the sterilized conditions, okay. So that is the bioproduction, clear. So remember this one thing, a simple step procedure is there, but remember what are the basic facts we needed, okay. It may be also considered the recombinant DNA technology, that is also the biotechnology, clear. So the question may be asked in any way to explain it, clear. So please take care of this one thing, keep studying, keep reading. In the next video we will learn about the application of the biotechnology for the welfare of the human beings. Till then, bye bye and see you later. Thank you.